So in this talk we will see the need for statistical experimental design in experiments with animals in preclinical studies. So we will see how to do it well and which are the pitfalls that are associated to this statistical experimental design when you try to do it. So typically you are interested in a particular treatment, let's say, and then you will have two groups and you make a number of observations for the control group, you do a number of observations for your treatment, and then you compute the mean of the control, you compute the mean of the treatment, you see that there is a difference, and then you want to see if this difference is statistically significant compared to the variability that you observe in each one of the groups. And there are statistical tools to do that. The most well-known and the typically used is a hypothesis test. And the hypothesis test, you try to decide whether the null hypothesis is true or you should reject it. So the null hypothesis is that there is no difference between the two treatments. So the mean in the two groups is the same, and that is either true or it is false. So that is the state of nature. Then you collect your data, in the analysis you want to decide whether based on your observations you should reject the null hypothesis. So if you reject it, there is a true difference between the two groups, or you should not reject. So if you fail to reject the null hypothesis, and then for the moment you are bound to believe that there is there is no difference, or if there is a difference, you have not been able to detect it. So if the null hypothesis is true and you reject it, then you have done the correct decision. If the null hypothesis is true and you don't reject it, then you have also taken the correct decision. But then there are two possibilities of errors. One is that the null hypothesis is true and you reject it. That is called a false positive, or type 1 error in the technical literature. Or the null hypothesis is false but you fail to reject it. So there is a difference, but you don't recognize this difference, and that is called a false negative. So we have true positives, true negatives, false positives, false negatives, and then we can put probabilities to each one of these cases. The two most important ones are the false positive rate and the false negative rate. So which are the probabilities with which you accept the false positive or you incur in a false positive and in a false negative. And 1 minus the false negative rate is called statistical power. The false negative rate, this one is called beta also, and the statistical confidence is 1 minus false positive rate. So this is typically alpha, and it is 1 minus alpha is the statistical confidence. And these alpha and beta, they have to be understood as this. So it is the probability of rejecting the null hypothesis when the null hypothesis is true, or the probability of not rejecting the null hypothesis when the null hypothesis is false. Okay, so this is what you will do when you perform your experiment. But how can you prepare for that? And you can prepare for that by calculating the sample size. So let's say that you foresee that the change that you want to detect is about 1, and the standard deviation of each one of the groups is also 1, and then you decide which are the false positive rates and the false negative rates that you want to admit to take. And actually this should be beta, not 1 minus beta. And then how many tails? So in this hypothesis test that you do, how many tails you will consider. So you will consider 2 tails or just 1 tail, and then if you consider in 2 tails, you will need 27 individuals animals per group, and this calculation is performed by comparing the expected distribution of the critical or not the statistic that you want that you will test at the end, if the null hypothesis is true in red, and if the null hypothesis or the alternative is, hypothesis is true here in blue. And then these areas are the alpha and betas. So you see that if what you want to detect is of the same size of the noise, then and you want to be very accurate, then you need more animals. And then depending on what is your test, so if you want the treatment to be always better than the control, not only being different, then you have to make a 1-tail test, not a 2-tails test. Then you see that there is a small reduction of animals, about 15% or 20% of the animals. So you could avoid doing experiments with 10-15% of the animals. Also, if you are looking for bigger changes with the same accuracy, then you can drastically reduce the number of animals to a quarter of the original size. And also, if you want to admit some higher false negative rate and not being so precise, you can also save some more animals. So you see that the number of animals that these you decide that before doing the experiment can drastically depends on what you are looking for, what is the variability that you expect in your experiment, and how accurate you want to be. And here you have this dependence. So depending on what is the sample size that I want to, not the sample size, the effect size, so the difference between the two groups that I want to detect, I may need fewer animals or more animals. If I want to detect very subtle changes, then I will need a lot of animals for that if I want to do it precisely. On the other extreme, so let's say that we always do the experiments with a fixed amount of animals, let's say 8. So with 8 animals we are somewhere here, so we can detect relatively big changes. Does it mean that we cannot detect smaller changes? Yes, we can do. So here you see these big changes, they can be caught with a high statistical power with only just 8 animals, but smaller changes, they can also be identified, only that with less power. It is much more likely that you will lose these very small changes with only 8 animals. So with only 8 animals, you will never be sure if this is a small change, not never, but when you are only 25%, 25% of your experiments, you will be able to say this result is not by chance. I would reject the null hypothesis. The sample size, you can also think of it in this way. So there is a linear cost. Here it is expressed in dollars, but it is a cost associated to performing an experiment. So let's say that you have a small experiment and a big experiment. So the big experiment, you will spend more animals, more time, you have to maintain those animals, you have to feed them, and you have to buy the treatment. So it is more costly. But then there is an advantage with large sample sizes, that is, the probabilities of taking incorrect decisions are smaller. So we have seen it before. If the sample size is bigger, then I will be able to detect smaller changes. So if the treatment is really useful with a big experiment, I will be able to detect it. If the treatment is not useful, I will also be able to detect it. But if I have a small sample size, I may have high false positive, and I will have high false positive and false negative rates. So that means that I may miss useful treatments, or I may continue with an useless treatment simply because I was not able, with a small sample size, to detect that the treatment was good or bad. So if the treatment was bad and I continue, then that is the cost associated to the subsequent experiments. And if the treatment was good and I miss it, then there is a cost of opportunity that it is more difficult to quantify, but somehow we see that it decreases with the sample size. So the total cost associated to an experiment is somehow the sum of these two costs, and there is a region that is optimal. So this optimal region, it is also interesting to see, to understand that the sample size is somewhere around, in this experiment that I have drawn here, it is somewhere between 20 and 30, and it is not that it is 27 and not 26. So in that region, between 20 and 30, you have a good compromise between being able to detect what you want to detect and not spending too much on the experiment, not only in terms of money, but also in terms of animal lives. So in reality, what happens is that the sample size is related to 4 other variables. So it is related to the effect size, so the relevance, what you want to detect, the variance, how variable each one of the groups is, and these probabilities of false negative and false positive that we somehow have condensed into this statistical significance and statistical power. So once you have fixed these 4 variables, you can compute the number of samples. And if you fix the number of samples, then each one of these 4 variables will adapt. Probably not the variance, but the other one, the relevance, significance, and power, will be adapted to the sample size that you had, as we saw here in this curve. Okay, so how when the sample size calculation will fail? So it will fail if the estimate of the sample variance is wrong. So if this sigma that we have to use for the calculation of the sample variance, it is overestimated or underestimated. If the distribution of the samples do not follow this, the hypothesis assumed by the statistical test that you are using, if you are violating the statistics in some other way. So for instance, you assume that the two populations have the same variance and they do not, or they have the same distribution and they do not, or if you think that after taking the logarithm of your observations the data is Gaussianly distributed and it is not, or there is something called a large sample approximation that normally in biomedical research is not important, but it doesn't apply to your problem. In biomedical research, typically with animals, this is not important. What if the sample size that I calculate is too large? Then I have to change something. I can try to change sigma, so I can try to improve the measurement procedure so that it is more repeatable, more reproducible, it has less variance. I may try to introduce some other blocking variables that help me to reduce the precision of the errors. So we will expand this idea a little bit more now. Then I may try to use a variable with better precision. This goes again in this direction of trying to reduce sigma. If my variable is not too informative, like categorical response, yes or no, I can try to substitute it by an ordinal, by continuous variables that have more information than categorical. I may try to identify covariates and blocking variables somehow also go in the same direction to try to reduce the uncertainty in the model. May try to reduce the confidence level, the test power, or try to repeat measurements on the same way as a way to reduce sigma. I may use paired sample methods rather than two independent sample methods. So for instance, if I can do that the same animal is its own control, so by making measurements before and after the treatment, or left ear and right ear, so I may reduce the sigma, the variability of my observations, and then my sample size will be smaller. When you do an experiment, you are trying to estimate what is the difference or some parameter of your distribution. In the example I gave you before of control and treatment, it was what is the difference between the two groups. There is a true difference. So let's say we would like to have experiments in which we have low bias and low variance. So let's say that my true difference is the center of this target. So this center is the true parameter. And when I do an experiment with, let's say, 10 animals, I get this value. And if I try it again, I get this value. And I get this value. All these values are very close to the center. So they have, or they are very close to the same point. So they have low variance, and not only to the same point, but this point is the center. That is low bias. So if I make the average of all my estimates of all my experiments, then I would have the true answer. We may be in a little worse situation in which the average is still the true target value, but our observations are more variable. So this would be the case, for instance, if you have a smaller sample size. And there are two other situations in which you really don't want to be. One is you have a low variance, but the average of your observations is not the correct one. This would be the case if, for instance, you are making your measurements incorrectly, or even worse, you have high bias and high variance. So bias typically invalidates all your conclusions. So you don't want to be biased by any means. Okay, and somehow this bias and variance is also related to the complexity of the model. So our model in the control treatment is just one parameter, that is what is the difference between the two groups. But you may construct more statistical complex models, and these more complex models, and these complex models depends on the number of parameters that you have to estimate. This is the case of regression, and this is also the case now in all these artificial intelligence tools. So if you have a lot of parameters, then you will have a high variance of your estimates, and probably you will have low bias. On the other extreme, if you have very few parameters, then you will have low variance and large bias. And there is a trade-off region where your model is well behaved, that is somewhere where these two curves meet. The problem with these two curves is that conceptually they are correct, but in practice they are very difficult to estimate. But somehow you have to play with your models and see in which region they generalize well, and this can be well done in many all statistical in all experiments. So you simply keep part of the data for validating your model, and then so you use part of the data to estimate the model, and part of the data to validate that what you have estimated is still correct. So we have already introduced the concept of blocking. So actually these animals should be assigned to the control and treatment in a randomized way. But if you suspect that there is a variable that may affect what you are observing, for instance, let's say that sex and age may affect your observations, then this randomization has to be done at the level of homogeneous groups. So you would separate male from female animals, then within each one of the blocks you would separate between young and old animals, and once you have homogeneous groups with respect to the variables you're considering, that is age and sex, then here you randomize. So blocking and randomization are really good friends in designing your experiments, because when you analyze the data, you're interested in the difference between the control and treatment. But this difference, if you don't use blocks, it could be obscured by the variability introduced by the sex and age. But because you have used blocks, now you can attribute what is the contribution of the sex and what is the contribution of the age to the final observations. So you can take that variability out, and then you can more faithfully compare what is the difference between control and treatment, independently of the variability introduced by these two variables, which may be much higher than the one of the treatment. So blocking is a very important tool when you design your experiment. You see that this blocking has to be done before doing the experiment. So you have to plan for blocks before doing your experiment. And this blocking is also somehow related to how we can attribute the different variability components to each one of the factors that affect our measurements. Here the factors were sex and age, but what if I have other factors? So these bring us to factorial designs. So they are extremely powerful tools to analyze what is the effect of a given variable, and this given variable can be your control and treatment. So for instance, let's say that you are interested in finding the optimal combination of number of meals and daily calories for a weight loss diet. So you have two possibilities for the number of meals, 3 and 5, and you have 3 possibilities for the number of calories during the day, and you make all combinations. You have 6 combinations, and you could analyze the data in a one way and another way. So just each combination is a different treatment, and I see if there are differences between all the treatments. And this could be done, and this could tell you treatment 4 is better than the rest. But this treatment 4, you don't know if it is because of the number of meals, because of the number of calories, or it is a mixture of the two. You don't know that. So simply by changing the way you analyze the data, so you change your one-way ANOVA analysis by a two-way ANOVA analysis. Now you can attribute what is the variability induced by the meals, by the calories. Are there synergies between the two? So having a low number of calories but with a high number of meals is particularly useful. And so you may do all these analyses and attribute what is the change induced by meals, by calories, by synergies. So these factorial designs are somehow a generalization of these blockings. So the blocks is also factor analysis. Also, you are used to listen this advice that you should keep all factors constant except the one that you are investigating. So let's try to follow this approach, this advice in this experiment. So let's say that you are studying amphibian water uptake. So you will take a couple of species, frogs and toads, that are kept in wet or dry conditions before the experiment, because they, depending on whether they were wet or dry, they may change the results. And half of the animals will be given a mammalian. So you are studying amphibians, but you are injecting a mammalian water balance hormone, and for the other half you don't inject anything, and you want to see if the after keeping the animals half an hour in water, how much water they have absorbed. So let's say that you have 24 animals, and that we will not study the interaction between factors. So you want to see what are the main effects. So what is the effect of the species? Do some species take more or less water? What is the effect of being wet or dry before doing the experiment? What is the effect of the hormone? This is actually the one that you are most interested in. And then there are some noise associated because not all the animals respond in the same way. So here what they are saying is the change in weight that I see for a given animal will be an overall mean plus something that depends on the species plus something that depends on the conditions before the experiment plus something that depends on the hormone plus something that depends on the animal itself. So we have three factors that are affecting these results. So let's change one variable at a time. So we have to define a kind of base condition. So the base condition for us will be frogs dry with no hormone, and then we want to study the effect of the species. So we will have toad dry with no hormone, and we have 4 animals in each one of the groups. And then with this comparison we can see what is the effect of the species, and then we change the condition, and then we change the hormone. In each one of the groups we have 4 animals, then we can analyze what is the effect of each one of the factors. But if we are asked what is the effect of the hormone in toads, we don't know because we haven't tested. So all we have tested is the effect of the hormone in frogs, but not in toads. So this design does not allow to answer many useful scientific questions. And also we may also notice that frogs try no hormone. It appears 3 times. So we have actually 12 animals for this one, while for the other groups we have only 4 animals, which is rather unbalanced. Actually we only have 4 groups. We have 3 groups here and 1 big group, but of animals of the same kind. So we may now rebalance the number of animals in each one of the groups. Because we have 4 groups, we can put 6 animals in each one of them. So now the numbers are a little bit more balanced, but still we cannot answer what is the effect of the hormone in toads because we don't have that group here. And then factorial designs, they try all possible combinations, all possible. So frogs dry no hormone, frogs dry with hormone, frogs wet with no hormone, frogs wet with hormone. So all 8 possible combinations. For each possible combination we have 3 animals. So you see that in these 3 designs we have 24 animals in total. So it is not only I will compute the sample size, it is how you will distribute your sample size between the different factors that may affect your results. Actually, the best design from the statistical point of view that you can have is this factorial design. So the fact that you have only 3 animals in each one of the group actually is not bad, because you will use all the groups to estimate what is the variability of your observations, and then for this because you have the 24 animals to estimate that variability, you will have a lot of confidence on your results. So actually the factorial design advice is hold all factors constant except those whose effect you are investigating. So those effects that you're investigating here, species, conditions, and hormone, should be varied as much as you can, as suggested by this factorial design. And also factorial designs are very flexible, so you can design them. Not all possible combinations must be taken. So for instance, let's say that you want to see what is the effect of applying two chemicals that you call E and O versus a control, and the chemical O requires a surfactant that you have two possibilities, and all these treatments, they can be applied into with two different sprayers that change the drop size. So maybe the drop size also have an effect, and you can also use different concentrations. So here you have our design. So our design will be, for the control we will use spray 1 and sprayer 2, and for the control it only makes sense to have this concentration 3, so we will make 5 replications of this combination. But then for the chemical E, you will also use the two sprayers, but now you will have two concentration, two replicates of 3 different concentrations. So you see here in total you have 12 observations, here you have 10, and then for the chemical O you will use the 2 surfactants, and then inside each surfactant you have 2 sprayers and only 1 measurement per concentration, and again here you have 12 measurements. So you see that if the experiments are balanced, the better. So if the number of observations in each one of the groups is the same, it is better, but it is not a catastrophe if they are not. So you can design your factorial designs in very flexible ways as shown here. And also factorial designs, you don't need to run all possible combinations. So let's say that you are interested in what is the production of antibodies in rabbits when you change different variables. So you may change the diet type, the temperature of the animals, the environment temperature, the humidity, oxygen concentration. You can change many things. You don't know which one of them will have an effect on the production of antibodies. And then if you want to do all possible combinations are 2 to the 7, because you have 7 factors, so you would have 64 combinations of factors. Do we really need to do the 64 experiments? So in the first approach, if I want to see which one of these variables is really important, then I can do these screening designs or fractional designs. So these screening designs tells you, no, you only have to do 8 different combinations with 2 animals in each one of the combinations, 2 replicates, and the first combination will be you have to associate 2 values. So you have already 2 values: standard show versus high protein diet. So let's say that this is plus, this is minus, this is plus, this is minus. So in the first combination you will use plus from the first factor, minus from the second factor, minus from the third factor, and so on. And this is one kind of design. The other kind of design, these are called fractional factorial designs, and again this is a little bit too technical, but the main message is you only need 16 animals, 16 in the fractional factorial, 16 also in the screening design. So with only 16 animals, not 64, and these 64 are the number of combinations. If you put 2 replicates per combination, you would have 128 animals. So with very few animals, you can identify which of the factors are really important, and then concentrate in a full factorial design just for those 2-3 factors that are truly important. Another way to design the experiments, or another thing that we have to consider, is blinding. So we are doing our experiments because we have a vested interest in the outcome of the experiments. we want to see some significant results in the treatments that we are designing. So if because of this interest, you might unconsciously do things differently when you are applying the treatment and when you are applying the control. And then this is particularly important when you have to make subjective measurements, like measuring behavior, scoring histological sections, or even making measurements. You may do the measurements more carefully when you have the treatment than when you have the control. So the best approach that you can have here is to blind the experiment so that the person doesn't know what he's applying and what he's measuring, he or she. Then the statistical mantra for experiment design: be control what you can, block what you cannot, and randomize the rest. So you can control the factors, you can control the treatments, you can block those variables that you cannot control. But for instance, you can control the sex, you can control the age, you can block the sex and age, and then randomize the rest. Randomize means randomize the position in the shelf, randomize the order of feeding, randomize the time of treatment, randomize everything that you can think of. I know people that even randomize the student that is making the experiment and randomize the pipette that they are using because they might be miscalibrated. Then, so this is how to do a statistical design right. How to do it wrong? How to do it wrong is these statistical pitfalls. Statistical pitfalls they are related to different things. These ones are related to the analysis. So being statistically significant, so let's say that you have a statistical significant result, does not mean that it is biologically relevant. So for instance, if you find a body temperature increase from 37 to 37.1 in 2000 animals, because you have so many animals, you have a lot of power to say these two numbers are different. They may be different, but it is not biologically relevant. There is no biologically relevant difference between these two groups, even if it is statistically significant. But the opposite is not true. So before being biologically relevant, it must be statistically significant. Otherwise, you are unsure if the difference you are observing is because of noise. So for instance, let's say that you have two groups and the temperature in the two groups are the difference is 1 degree, and so this might be biologically relevant, but you have only observed 3 animals. So how do you know this difference is just is a true difference, or it is just because of noise? So before being biologically relevant, it has to be statistically significant. The opposite is not true. Being statistically significant does not mean that it is biologically relevant. Then another pitfall that we may do when we analyze our data is p-hacking. So having this alpha, this false positive rate to 0.05, that means that in 5% of your experiment, that is 1 in 20 experiments, in which the null hypothesis is true, so there is no difference between the treatments, you think that because of the observation that you have had, you think that there is a difference. So, and that happens every 20 experiments, but you are also doing this every 20 experiments if you are measuring 20 variables and you are doing 20 hypothesis tests, or if you are measuring at 20 time points. So this 5% of significance is going against you. So because 1 in 20 you will think, on average, 1 in 20 you will think that there is a difference when there is not. And this is, if you're measuring many variables or many time points, this is called response selection. So you only report the one that is statistically significant. Or group selection. So if you divide the animals in many different ways, or combine the groups in different ways, or remove the animals after data collection because their results are not aligned with the expected outcomes. So every time you remove an animal, you are subdividing the data in different ways. Or monitor the p-value and stop the experiment when the results are significant. So you do your experiment with 1 animal, 2 animals, 3, 4, 5, and when the p-value reaches this level of significance, you stop, because there you're doing many tests. At every number of animals you are doing a test, and you risk this alpha false positive rate, rejection of the null hypothesis when it is true, and repeating the experiment multiple times until you find the p-value. So all this is affecting to group selection. Or you may change your hypothesis. So for instance, you try two tails test, two one-tails test, a non-parametric test, and an ANOVA. So every time you do a test, you have a false positive rate. So you have a probability of rejecting the null hypothesis when you shouldn't do it. Or including covariates that were not planned. So you include all kind of covariates in analysis, and then so temperature, the lab humidity, or whatever, and for every variable that you add, you again have the 5% probability of declaring a result significant when it is not significant. So this p-hacking, as the number of tests increases, and here all these situations, what they have done is increasing the number of tests. So as the number of tests increases, the probability of false positive detection increases. So if you do just one, it is 5%. If you do two, then it is 5% in the first test and 5% in the second test. And if you have 100 tests, then the probability of not rejecting, let's say that the null hypothesis is true, but because you have tested it in 100 different ways, the probability of not rejecting the null hypothesis in all of these 100 tests is very low. There will be one of them that will reject it just by chance. So if you do a lot of tests, the likelihood of the probability of rejecting the null hypothesis is very, very high. Then we may have dependent samples. So this is something that you can design from the beginning, how you will analyze your data. Don't change that plan, that analysis plan. Okay, so then also something that you can do from the very beginning is to identify whether I will have dependent or independent samples. So all the statistical tests, they assume that the data is statistically independent, and they don't account for the data correlation. There are ways to account for the data correlation, but the standard ways that we use in the labs, normally they don't. So if I have technical replicates, the right way of dealing with the technical replicates is averaging them, and then you have a single value associated to that animal. Or you do repeated measures at multiple time points of the same animals. So all these measurements are not independent. So there are correct ways, and we have already seen it. The animal is a block, and then I should measure what is the variability introduced by these blocks. And litters and cages, they're also blocks. Animals there, they share genetics, or they share the environmental influences, so they should be blocks too. We have a batch effect, so that is some other kind of blocking in which the animals or the measurements from a given week, they may have systematic difference from other weeks. So because of the experimental, because of the chemicals, because of the weather. So all these are blocks that I can try to estimate what is the contribution, their contribution to the final observations. Then I may fail to compute the sample size correctly, and a common misunderstanding is that the reduction in the 3 R's means using very few animals. And if you do that, that insufficient, those insufficient sample sizes will compromise the statistical power and the validity of your conclusions. We would be at the beginning of this curve of the coast, where we had a high probability of making incorrect decisions. Then if you use fixed size experiments, you always do the experiments with 8 animals, then you may underestimate or overestimate the required sample size. So the number of animals should be adapted to the variability that you expect, and also to the size of the changes that you want to detect. Then you may be too optimistic on the variance, so you may think that you have a lower variance that you actually have, and then that will increase the likelihood of false negatives, because you have underpowered experiments, so you will miss many good treatments. Or you may use an incorrect formula to compute the sample size. So let's say that you use the formula for the student t-test, that is the most common one, but later you will analyze the data using ANOVA. So the sample size that you have calculated for the student t-test is not the one that you need for ANOVA, or if you are analyzing proportions. So you should use the correct sample size formula for the type of analysis that you want to do. Then you might have planned from the beginning to use an incorrect statistical tool. So for instance, you may do a student t-test for non-continuous data, so that doesn't make sense from the theoretical point of view. Or you may decide to use ANOVA to analyze survival rates, which again they are not the right tool for that. Or you may not include all the factors and the covariates that explain part of the variability, and then you have all these factors and covariates that affect your measurements somehow confounded in your noise, and then you're lowering the statistical power of your analysis. Or you may try to do a full factorial when a fractional factorial design would be enough, and then that is a waste of resources and animals, and you are not gaining much from the point of view of statistical analysis. Or you may use a normal distribution when it is log normal. This is, for instance, very typical when you are analyzing virus titers or dilutions. The data is log normally distributed. When you do an ANOVA with the data without transforming, all the results that you get, they are invalid. And typically what happens is that they show to be non-significant when they can be significant. Then you may be using always a student t-test, which is a parametric test that assumes that the data is Gaussianly distributed when your data is not Gaussianly distributed, and then the p-values calculated by the student t-test are not correct. So you should use a non-parametric test to analyze this data, because you are not fulfilling the assumptions of the test. And you may have the opposite situation, when because you are afraid of using a student t-test, and then you always use a non-parametric test when actually the data is Gaussianly distributed. If the data is Gaussianly distributed, the student t-test is perfectly fine, and it will be more accurate, it will calculate better the p-values than the non-parametric test. The non-parametric test has less statistical power, because it doesn't know something about the sample. It doesn't know that it is Gaussian, and it is not making use of that knowledge. But the student t-test can benefit of that knowledge if the data is truly Gaussianly distributed. So they are more powerful. Then many times we do some kind of regressions, and there are a number of statistical pitfalls associated to regression. One of them is extrapolating. So regression is only valid within the range of values we have observed. Going beyond those values, you may get incorrect predictions, and sometimes you also get valid predictions, but you don't know. It is valid only inside the range you have observed. Or you smooth the data before making the regression, and then you are distorting all the p-values, all the r-squares. So you might be overconfident on your prediction probability, and doing an overfitting of the model, so including too many predictors that are capturing the noise rather than the actual relationship. This is we gave an example before when we had these models with many parameters. That is called overfitting. Then ignoring non-linearity relationships. So depending on how you design your regression, you may account for linear or non-linear relationships. If the data, if there is a non-linear relationship and you don't account for it, then you are distorting your conclusions. Or this is the opposite of this one. So you may omit variables that are useful to make predictions, so to predict part of your observations, but you are not including them, and then you have incorrect conclusions, misleading coefficients, because the rest of the coefficients of the rest of variables will try to account a little bit for the variable that you have omitted. And another problem is when you include many predictors, and these predictors are collinear, so they are highly correlated between each other. So these highly correlated predictors, so let's say that you include an extreme case, you include the weight of the animal in pounds and the weight in grams. So those two weights, you have two variables, but they are highly correlated. So and this will inflate the standard errors and make it more difficult to isolate what is the effect of the weight. You don't care if it is measured in pounds or in grams. Then misinterpretation of causality. So regression identifies association, not causation. And if you interpret the results, the regression results, as causes, you may be making false claims. Then having from the very beginning an inadequate sample size. So if you have not designed the sample size for making the regression, and if you have a small sample size, then all your coefficients are rather unstable, and if you repeat the experiment, you may have relatively quite different coefficients for your relationships. Then all the p-values calculated by regression, they assume that the residuals, that is the difference between your predictions and your observed values, are Gaussianly distributed, independent. So if this is not the case, then the p-values and the confidence intervals that you compute from the regression coefficients, they are incorrect. Then if you include in the regression variables with two different scales, so let's say that one variable goes from 0 to 1, the other one goes from 0 to 1,000,000. So these two factors that are together in the same regression as predictors, the one from 0 to 1 will be completely disregarded with respect to the other one, because it doesn't have enough weight, because it is only changing from 0 to 1, the other one from 0 to 1,000,000. So these different factors with different magnitudes, they hinder the interpretation and the numerical stability of your regression. Then another pitfall, and this is again from the very beginning, from the experiment design point of view, it is planning to repeat the experiment twice, for instance, if you want to see what is the effect in males and females, or 3 times. So 3 times just to give a statistical significance. And statistical significance has to do with bias and the variance that we were talking before when estimating parameters. For the variance, the sample size calculation that we do perfectly addresses the variance. The bias cannot be addressed by that, so that is why you want to repeat the experiment. So let's say that you have a miscalibrated scale in this week, but and that is distorting your observations, and that is why you want to repeat. Maybe when I repeat the experiment, I have calibrated the scale correctly. I didn't know it was the miscalibration of the scale, so I have calibrated it correctly, and then I don't have incorrect results, biased results. So, but rather than repeating the experiment, let's say that you decide that your sample size should be 39 individuals, animals per group. So instead of repeating the experiment with 39 3 times, what you should do are mini experiments. So you divide your experiment into 3 mini blocks of 13 animals each, and then if in one of the weeks you have a problem bias results, you will be able to identify, because the other 2 weeks are given consistent results with each other, and also you will be able to identify what is the bias, and you will be able to correct for it. So it is better to rather than repeating, and the same applies to sex, so rather than repeating the experiment 3 times, it is better to make mini experiments and treat those mini experiments as blocks, and then analyzing all the data together. And then there are some other pitfalls associated to our psychology and attitudes. So you may have often heard, I have a long experience doing this, and then this overconfidence can result into outdated practices or misinterpretation of the data, or it is too theoretical and cannot be applied in real life. So it overlooks that you may reduce the number of animals or interpret better your data, especially if it is complex data. It is too complex. So if you think that using statistics is too complicated and you don't even try to do it yourself or help or look for help, you may be missing opportunities to reduce the number of animals, and you're turning your experiment suboptimal due to a misuse of the data. Or that's great, the statistic is great, but I don't know how to do it, so I will not apply it in practice, so it is the same. Or thinking that the statistics is just for statisticians. So this discourages interdisciplinary collaboration. You may look for some colleague that knows how to use these tools, and you are preventing yourself from leveraging tools that might be very, very useful in your work. Thinking that you can prove anything with the statistics, this is a cynical attitude, the attitude that dismisses statistics as inherently biased or manipulative. So at the end, it is undermining distrust in the use of tools that help you to take decisions based on the data that you have. Or if it is not significant, it is useless. So not significant results. Maybe you have an underpowered experiment, so you don't have enough samples to prove what you are seeing, so it is not statistically significant, but you see the trend, and then it is a good suggestion for repeating that experiment now with a proper sample size, so that you can benefit from the insights from a previous experiment. Thinking that okay, I'll throw the data into the software and then it will manage it. So this blindly trusting the software is somehow uh, risks misinterpreting the results and poor decision making, because uh, the software is nothing more than a calculator. If you use the calculator incorrectly, the results that you get, they have been correctly computed, but they are not the computations that you wanted to do. So you may have uh, you may misinterpret quite a lot of your results. Thinking that you only need the data for p-values, it is an oversimplification of the tools. So you may get more from the tools if you try to understand what they tell you, and you may be ignoring model assumptions or any other aspect of the tool that might be critical for your analysis. Thinking that statistics is just common sense with math, in a way it is somehow correct, but it also trivializes the discipline, and you are ignoring the expertise that you require to apply the statistical tools appropriately. If you don't trust your data, then there is no. So I don't trust the data, so why should I bother with the statistics? So the analysis tools can be also can tell you whether you could trust your data or not. Making the experiments and you'll figure out how to do the statistics later is a poorly designed study. So we have seen already that you should know from the beginning what is your analysis plan, and actually you should compute the sample size according to this analysis plan. So depending on how you will analyze the data, you will compute the sample size in a different way. Thinking that using the statistics correctly it is too time consuming leads to cutting corners in the analysis and increasing the likelihood of errors. Having overconfidence also is bad, so everyone knows the results, why run the analysis. So this this view fosters confirmation bias. So you only will pick up the results that confirm your expectations, and it will weaken the scientific credibility. Statistics isn't necessary for small data sets. Actually, statistics in the small data sets can quantify especially the uncertainty that you have in your results, because if you have very small sample sizes, then you have a lot of uncertainty, and statistics can help you to quantify how much uncertainty you have. Or I'll just follow what others did, and this is blindly copying statistical methods from similar studies without understanding if they are suitable or not for your case, can lead to invalid conclusions. And saying it is all subjective anyway, so actually it's dismissing the objectivity that a good use of statistics can bring. So and that perpetuates biases, undermines decisions taken on your observations. So statistics are very objective, and thinking that you should not use them is again going against you. So in conclusions, if your experiment is important, it deserves to be correctly designed from the very beginning, and that you use the best tools that you have at your disposal to do the analysis and to do the experiment. And that is obvious from the experimental point of view. So you want to use the best chemicals, the best devices, the best equipment, but also for the data analysis. You want to use the best statistical tools that you have around. Also, the research animals that participate in your experiments, they deserve that you design your experiment correctly, and that you use the best tools at your disposal. You don't do the experiments in any way because they are suffering for giving you an answer, and you should be ethical to them. And the funding agencies of the general public that are paying your experiments, they deserve that the scientific funds are used judiciously. You don't need to do the calculations by yourself. So if you don't know how to do them, team up with someone who can do the calculations for you and the design for you, and he's appropriately recognized in your experiment. He's part of the experiment, and he should be part of the publication at the end of these results. And be open-minded. So if you are looking for help, try to understand the statistics basics. You don't need to understand the very core details of the statistics, but try to understand the minimum, and so that you can talk to the statistician. The statistician should be the same, so he should try to understand the biology basics, not the very core details of the experiment that you are performing, but you should be open-minded to be able to talk to the other person with a different background. And with this, I'm done. I would like to recommend you this book on the statistical experiment design, especially for preclinical studies. Actually, the statistical experiment design applies to any discipline, but here all the examples are in preclinical studies with animals, and you will recognize many of your situations there in the examples that you will find in that book. That's all. So thank you for attending.